turn the podium over to Professor Sagar Khare. He's an associate professor in the Department of Chemistry and Chemical Biology, a resident member of the Institute for Quantitative Biomedicine, and he's also the head of the Institute for Quantitative Biomedicine graduate program. And today, Sagar is going to tell us about the structural biology and evolution of the Papain-Like Protease from within NSP3, a large multi-domain protein, part of the SARS-CoV-2 proteome. David did his postdoctoral work with David Baker at the University of Washington, and is one of the authors of the suite of Rosetta programs, which have proven to be so powerful for understanding protein structure and protein energetics. And Sagar is the co-chair of this event. He'll be chairing the second half and providing the closing remarks. And I want to thank him again for working so closely with the PDB team this summer to make our summer research experience for a dozen students such a rich and valuable one, and one that, as you'll see, is producing some quite important findings regarding the evolution of the COVID-19 proteome since the pandemic began in December, and providing information all the way up to nearly the end of June. So thank you again, Sagar.
Thank you, Stephen, and thank you everyone for joining us today. I hope you can see my screen, as a full screen presentation. If not, let me know.
We're good on both screen and volume, Sagar.
Okay, perfect. Thank you. The two most important questions during the pandemic: can you hear me and can you see my screen? I'm glad those are answered. So I'm going to tell you a little bit about what we know. We as collective scientific community know about SARS-CoV-2 Papain-Like Protease, or at least what I have been able to gather, and some of the work that we have done in trying to understand how this protein, this important protein, has changed during the pandemic. So I'll first tell you about the role of this protein in the virus life cycle. I'll tell you a little bit about the enzymology and structural biology that has been done. As David just mentioned, it's been just remarkable how quickly structures have been solved and really knowledge has been generated. So I'll tell you a little bit of that. Of course, it won't be an exhaustive overview because of time considerations. And then I'll tell you a little bit about how this protein and other SARS-CoV-2 proteins have evolved, as an overview, and how things have changed and what implications are for inhibitor design, drug design, and drug resistance, and so on. So as David mentioned, the viral proteins are expressed as a long polyprotein, which requires cleavage by two viral proteases. And Papain-Like Protease, or PLpro, is one of those two proteases that makes cleavages at these indicated sites by blue arrows. So effectively, it liberates three proteins, including itself. And then once it has liberated these proteins, 3CL-Pro will make the remaining cuts, and the viral assembly will go on. So obviously, as you can imagine, these cuts are necessary for viral maturation, and therefore this is an important protein whose function needs to be understood and is also a valid drug target. Now, like many viral proteins, it does many functions, and in particular, it does some very important functions related to shutting down innate immune sensing and response in the host. So I told you that PLpro, which is part of this large protein NSP3, is going to cut the viral polyprotein, but it will also cut these small marker proteins called ubiquitin and ISG15 from host proteins. And the host proteins that it targets are many in number as well as in their function, but the effective response is that it shuts down type 1 interferon type responses and interferon stimulated gene expression. So this essentially dampens the innate cellular immune sensing and signaling that will send a signal from non-immune cells to immune cells, as well as cause changes in the metabolic state of the cell. So PLpro is therefore a very important enzyme that has both these very important functions. And therefore, in order to understand this enzyme, people have looked at both of its suite of functions. So currently in the PDB, there are 22 apo and co-crystal structures of PLpro. If you can go to a page which lists what PLpro structures are available, and you can see that there are 22 of them. This is a little bit more about this enzyme. This is a papain-like enzyme, and it has a cysteine, histidine, and aspartate catalytic triad, which is well known. Overall, its structure is actually closer to human deubiquitinases rather than chymotrypsin, which is the 3C-like protease is more similar to. Before we knew the structure of SARS-CoV-2 PLpro, which became available in May, I think, we relied on the structure of the SARS-CoV or SARS-CoV-1 enzyme, which was solved back in 2006, because the amino acid identity between CoV-2 and CoV-1 is about 83%. And then after the structure has been solved, it was found that the RMSD was very low, meaning that the two structures are nearly identical. If you look at the structure a little bit, there are essentially 3 or 4 regions. So first you have the fingers, the palm, the thumb domains, and the ubiquitin-like domain. And the active site is located at the interface of the palm and the thumb. And you can see that here, this is a representation of the active site cysteine, which sits at the base of this alpha-4 helix. So this alpha-4 helix is going to be important. So I would like you to just make a note of this. And also this alpha-2 helix, which is at the interface of the thumb and the ubiquitin-like domain. Both of these alpha helices are going to feature prominently in the rest of the talk. So the first thing that I would like to highlight, which I think is very important and interesting, is that even though the structure of and the sequence of SARS-CoV-1 and 2 are very similar, a recent study published in Nature a couple months ago found out that they have some very important and striking differences. And the first one of them is that the SARS-CoV-1 enzyme prefers ubiquitin substrates. I told you that it can cleave off both ubiquitin and ISG15 off of human proteins. And it turns out that SARS-CoV-2 prefers ISG15 substrates. And this was illustrated both in the binding of these proteins with ubiquitin and ISG15, as well as their catalytic activity. So if you look at these data here, SARS-CoV PLpro, the Kcat over Km, which is the catalytic efficiency of this enzyme, has a higher catalytic efficiency against diubiquitinated K48 lysine linked substrates, whereas SARS-CoV-2 has a higher catalytic efficiency against ISG15 containing substrates. Not only that, if these authors went on to do pull down experiments to identify the interactome for both of these PLpro molecules, and they found that there are significant differences in what is pulled down or what these PLpros are interacting with. So you can see here that the amount of ISG15 that is pulled down by SARS-CoV-2 PLpro is significantly higher compared to SARS-CoV-1, where there is a depletion of ISG15 in contrast with ubiquitin, where SARS-CoV PLpro is going to pull down ubiquitin, meaning that it's going to bind to very different proteins, and that is dictated by its binding to ubiquitin and ISG15. So the interesting question is that even though the active site is 100% conserved, what leads to these kinds of effects? And one possibility is that there are different binding modes and different binding strength as a result of small but significant changes on the surface of these proteins. And those types of differences were indeed revealed by crystal structures that have been solved. I should say that the biological significance of this interactome difference is not known, but this is, again, one of those open questions which may help address perhaps the differences in the initial innate immune response to SARS-CoV and SARS-CoV-2. But of course, that's a long leap to make, but I think that these data are extremely tantalizing and open a lot of important and interesting questions for the field. So the crystal structures of SARS-CoV-2 PLpro and its complex bound to ISG15 has been solved. And you can see the structure described in this Nature paper earlier this year. And what you can see is that, I told you that there are those two helices that are going to be important. So here's that S2 helix that I told you about, right next to the ubiquitin-like domain. So that participates in binding to one of the two domains of this ISG15 protein. And you can see that the ISG15 protein, if you look at the structure of this protein by itself, this second domain, which comes in and latches on to this S2 helix, is apparently in a different state in the apo structure. So it has to rearrange in order to bind to this particular S2 helix. And so this rearrangement from the apo state was observed. And there are key interactions that I'm not showing the details of, but you can see them in this paper with both the N lobe or the N terminal domain and the C terminal domain of this ISG15 protein, right? So this was illuminating, and then immediately one can see why there might be differences in the preference for ubiquitin versus ISG15. So here, although the structure with diubiquitin and SARS-CoV-2 is not known as far as, to my knowledge, but we know the structure of SARS-CoV-1 bound to ubiquitin, to diubiquitin, and SARS-CoV-2 bound to ISG15. So diubiquitin is essentially two ubiquitin domains, and each of those domains is shown in cyan and pink here. And the MISG15 protein from the previous slide is shown in red. So what you can see immediately is that there are different binding modes for both the lobes of ubiquitin compared to ISG15. And the key interactions for ISG15 includes these sites S2 and S4 helices, even though if you look at the structures of the two PLpros, they are nearly identical. You can hardly distinguish them. The RMSD is close to 1 angstrom. So that sheds some light, although of course, much more remains to be done. And I think this is going to be very exciting in trying to make connections, especially with the biological implications of these structures. So apart from substrate bound structures, there are several covalent and non-covalent inhibitor bound structures. So GRL 0617, which is an inhibitor that's known to inhibit viral replication, although it's not an excellent inhibitor because it is required at high concentrations. But nonetheless, this inhibitor can shut down viral replication in infected cells, and therefore provides proof of principle for research groups who are working really hard to develop novel inhibitors. And we're working on that with the lab of Nilgun Tumer here at Rutgers. There are also covalent inhibitors because this is a cysteine containing enzyme. There are electrophilic inhibitors that are based on peptides or peptide-like molecules that people are developing that covalently target and shut down the enzyme irreversibly. And there are a couple of examples of those also. Those structures have been deposited. And so with all this structural information in hand, along with Stephen and the PDB team, earlier in the summer, we decided to think about how the entire proteome of SARS-CoV-2 is undergoing changes and what its implications might be for therapeutic design, as well as for fundamental virology and other therapeutic interventions that are in the pipeline and coming out. So I'll tell you a little bit. We discussed this in great detail yesterday, so I won't go into too much detail. but I'll just tell you a little bit of what we did, how we did it, and what we found. So we started by looking at a protein that Stephen is going to tell you in detail about, which is NSP5, and looked at, analyzed all the sequences that were available and the structures that were available with a team of various participants in a IQB summer boot camp. And all of that information is available. So if you're interested, you can go and do it for yourself. And then we had a group of undergraduates and a very talented high school student who joined us for the rest of the summer to perform analysis of how the viral proteome is changing. For about a month and a half, we worked together and analyzed about 50,000 proteomes. So we obtained these data from GISAID. We analyzed 50,000 proteomes. And for each of the 29 viral proteins, we classified the changes as single, double, triple, etc. mutants. And we performed structural modeling based on Rosetta, performed Rosetta energy calculations that I'll tell you a little bit about. And this work involved a large number of people from the PDB team, as well as all the people in my lab contributed to this, and of course the summer interns who participated in this work. So I'll just briefly summarize what we found. So the first thing that we found was that most mutations arise as a result of single base changes in the RNA. So here you will see substitution matrices where you have the native amino acid on the x-axis and the substituted or the mutant residue on the y-axis, and they're grouped according to their chemical type. So you can see that hydrophobics change a lot to other hydrophobics, as might be expected, because these sequences are from viruses that have survived. So we expect that all their proteins are still functional, and therefore these changes are conservative. But a large number of changes are also non-conservative. And in most cases, we can explain them by the fact that the structure of the genetic code makes some of these changes much more likely than others, even though they are non-conservative. In terms of energetics, what we found was a very interesting bi-Gaussian distribution corresponding to the energetic effects of these mutations. So you can see that this is a histogram of the calculated delta G values, the changes in thermodynamic stability. And you can see that they fit really well to a skewed Gaussian or two Gaussian peaks, one corresponding to about 1.3 Rosetta energy units, which is similar to a kcal per mole. And the second peak corresponds to about 6 kcal per mole. And if you look at where these peaks are coming from, you will see that the first peak largely comes from surface changes, which, as you might expect, are expected to be really just moderately destabilizing, whereas the second peak comes from a mixture of boundary and core mutations, which are in the constrained environment of the core are likely to be more destabilizing, but can occasionally also turn out to be more stabilizing by packing better. And in fact, we were not the first ones to find this type of distribution. Others, including Danny Taufik's lab, Eugene Koonin's lab, have found these type of distributions in experimental data, as well as calculated stability changes upon mutations across proteomes. So these distributions that we found reflect a sort of universal constraints on mutations in the different protein microenvironments, given that proteins are globular, and depending on where the mutation is, that's going to have a different kind of effect. Okay, so I'll tell you now a little bit about this analysis for PLpro, and these are some of the structures that we looked at. So here we have the apoenzyme structure, we have a covalent inhibitor bound structure, a non-covalent inhibitor bound structure. This is the GRL 0617, as well as the substrate bound structure that I've walked you through. Most of our analysis was conducted on this apoenzyme structure, but we have used the results to sort of interpret some likely possibilities for how these mutations are going to affect these bound structures as well. So first, we analyzed about 37,000 viral proteomes, and 97% essentially had no changes in the papain-like protease. This is expected because coronaviruses have proofreading machinery, an exoribonuclease that corrects errors occurring during replication, and therefore the number of changes that occur are generally very small. So 97% had no changes. Nonetheless, we still had 225 unique sequence variants. About 215 of them were single mutants, 10 were double mutants, and there were no higher order mutants. Most of the mutations were non-conservative because they were likely occurring mostly on the surface. And in fact, about 89% of the mutations were on the surface or the boundary, and only a small number, around 10%, were in the core. Importantly, there are no active site mutations, suggesting that this protein's active site machinery is conserved in order for this virus to survive, and therefore this would be a good target for therapeutic drug development. If you look at the stability distributions, again, similar to the entire data set, we can see that the two peaks can be well explained or are similar to the peaks that are observed for surface, boundary, and core residues, respectively. So now let's look at a few different mutations that I would like to highlight. So first, this is the crystal structure of an inhibitor-bound PLpro, and I've also superimposed the ISG15 substrate structure on top. So what you can see is that on the right, this is the ISG15, this spaghetti-like thing is the ISG15 backbone that is going into the active site, and this is the inhibitor bound. So one of the first things that you can notice is that this inhibitor, in order to gain traction in the active site, has to make contacts with some other elements of the active site. So it's sitting against these two prolines in the active site, whereas the substrate is a little bit further away. However, it turns out that these prolines have indeed changed from prolines to larger residues or sometimes even smaller residues. So they've changed to a leucine, glutamine, and serine, and the second proline has also changed to a serine. So what that might mean is that these mutant proteins will have a different interaction profile with the inhibitor. They may bind the inhibitor less well compared to the wild type, but given that the substrate is farther away from the sites of these mutations, that may have no impact on substrate binding. And those are the types of mutations that can get fixed as drug resistance mutations. So modeling these mutations in three-dimensional space can be useful because one can, in principle, prospectively identify mutations that may cause drug resistance. So we're planning to make these mutants in our lab and indeed look at this type of inhibitor binding and see how it is, if it is indeed effective. There are several mutations. There are about 20 USVs that occur in the key S2 helix that I told you about, which is at the interface with ISG15. And some of these are listed here. I would just like to point out one of them, which is F1632S, which this phenylalanine in the S2 helix finds, nestles into this nice hydrophobic cavity on ISG15. And in this particular mutation, which in the apo state, remember, is found to be mildly destabilizing, may have different ISG15 binding properties and some of these others as well. And you can imagine that given the difference in the interactome of SARS-CoV-1 and 2, the effects that mutations have on ISG15 binding versus diubiquitin binding, for example, may have a profound effect on the type of interactome remodeling that these viruses, these mutant viruses, might be doing. So I think that these are would also be very interesting mutations to further explore. And then finally, there are also some destabilizing mutations. So for example, in the S4 helix, which is the site where ubiquitin and ISG15 both bind. So for example, there's mutation S1733 glycine, along with 8 other USVs that may lead to some kind of secondary structure destabilization and may again remodel the interaction as well as enzymatic properties, but all of that remains to be seen. So I think that these mutation data have provided us with really interesting hypotheses and may help in really understanding how the virus works and interacts with the innate immune response. So to summarize, what we learned is that first, that thankfully mutations are infrequent, meaning that this active site represents an attractive target for drug discovery. We know that there are several binding partners and several mutations as well as in the binding interfaces, which raise important considerations, in my view, for drug resistance as well as the biological function of PLpro. And the PLpro co-crystal structures in the PDB are a growing data set that are continuing to facilitate basic biological and structure-guided drug discoveries. So these are the people from my lab who, each of whom participated in this activity that I mentioned. And this activity has been spearheaded, was started by Joey Lubin, a graduate student in the lab who is now continuing with this work. And thanks again to the PDB team. And thank you for your attention. I'll take a question or two as well. Thank you.
Thank you, Sagar. So as before, I will accumulate questions in the chat. Please type into the chat if you'd like to ask Professor Khare a question. So while people are collecting their thoughts and typing into the chat, Sagar, could you talk a little bit more about the some of the challenges that you faced you and Joey and your team faced, because they were not insubstantial, in doing the energetics calculations? It was not like falling off a log, was it?
Yeah, no, I think it was far from that, and I'm sure that the people in the team can actually speak to that, with much more immediacy. But I think that there are two types of challenges. One was, dealing with the quirks of, PDB files, which is, people deposit with different type of numbering. There are, based on the crystallization conditions, there are different ligands, and so one needs to identify the right set of things to model. And the second set of challenges is how to make models, and so those are sort of the modeling program associated challenges, as well as making some decisions about what states to model proteins in. So, for example, I think David mentioned that sometimes we don't know what the oligomeric state of the protein is, so we have to make some assumptions. So we did a lot of that. Sometimes we don't have a structure, so we have to make a homology model, and then there are issues about how much to trust the homology model. Sometimes you have to make ab initio predictions if homology models are not known. So there is all of that quality control, as well as decisions to make. And then there are how to model membranes, how to model metal ions, how to model nucleic acid protein complexes, how to model oligomeric proteins. So there are lots of these decisions, and it was a really challenging modeling exercise, and the way we solved it really was by a lot of collaboration. So everyone sort of worked on different parts of different proteins, and we had a central sort of repository where we kept updating and modifying a script that ultimately we think is kind of generally applicable and useful. And we'll release all of our models and the scripts to the public, hopefully in a week or so, when we submit a manuscript describing these results.
So Greg Critchlow asks, proteases must be active in the polyprotein state because they liberate themselves from the polyprotein. He goes on to, which is true, he goes on to ask, is PLpro able to cleave ISJ15 bound proteins while in the polyprotein, or does it need to be free of its neighbors?
I think that's a great question. I don't think we know the answer to that question. At least, I haven't come across the answer. I do seem to recall that PLpro can cleave both in cis and trans. So, it doesn't have to necessarily just cleave itself to liberate itself. It can also cleave a PLpro on another chain. But whether it can still cleave ISG15, I don't know.
I suspect somebody is doing an experiment right now to find that out.
Yeah, I think it's very interesting, and somewhat under investigated overall. how viral polyproteins, what they look like and how they function, and how their functions are affected. And I think those are really fascinating questions.
They absolutely are. And there's some very influential and important work going on here at Rutgers involving Eddie Arnold and
Yes, yes,
Jason Kalber
yes.
on the HIV polyprotein.